Thank you, Jay. Thank you, Jay. Thank you, Jennifer, and the rest of the Alex Kentsis Foundation. As Jay mentioned, I've been supported by the foundation for many, many years, and it's safe to say that without it, my research would not be what it is today, and I would not be able to share these ideas that I'm about to tell you about. And the question that's been fueling me and many, many people in the field, of course, is why do young people get cancer? Otherwise, healthy children, why do they get cancer? And if you look at the epidemiology of all of human cancer across children and adults, the incidence of cancer rises steadily as we age, as shown on this curve. And the basis for this steady increase in cancer with aging is thought to be the accumulation of mutations in our DNA, sometimes due to exposures in the environment from mutagens, sometimes due to errors in DNA replication that sustains all of our normal tissue development. And indeed, studies across many cancers support this notion. But of course, it is not really sufficient to explain why younger people would develop cancers due to these exposures when they have not lived long enough to accumulate errors in DNA. Now, of course, there's a very well-recognized cause of childhood and early onset cancer that has to do with the predisposition towards cancer, usually due to the inheritance of mutations in genes that encode factors that regulate genome stability and DNA repair. Some of these are well-recognized cancer predisposition syndromes, but current estimates of this in both children and young adults ranges somewhere between 8 and 10% of individuals, meaning that the majority of children who get cancer do not have a recognizable cancer predisposition syndrome or mutation. Now, of course, that's an underestimate, and in the years to come, we will find more such cancer predisposing mutations, but I would posit to you that even when we do, they will still not explain the majority of childhood cancers. And this increased peak of incidence in childhood cancer has been puzzling the field for many, many years. And there is one common form of childhood cancer that gives us a clue. And these are lymphoblastic leukemias. These are cancers of the blood, of the developing immune cells, B cells and T cells, that of course are part of the normal adaptive immune response that's essential for vertebrate immune system development. And there it's now very clear that the majority of the mutations that cause these leukemias, chromosomal translocations and deletions that produce oncogenic fusion genes and inactivate tumor suppressor genes, are mediated by the RAG1/2 recombinase, the DNA recombinase that's part of normal somatic diversification in the immune cells, that for reasons that we still don't fully understand becomes dysregulated and creates these mutations in the developing B cells and T cells, ultimately leading to their transformation and leukemia. Recent work, some of it from my lab together with the Target Consortium and Alejandro Gutierrez, points that in a large fraction of acute myeloid leukemias in children, the same process is responsible for the production of leukemia oncogenes, suggesting that the majority of blood cancers in young children, again, are due to the dysregulation of this essential developmental somatic DNA rearrangement process. And it begs the question whether similar processes are responsible for mutations that occur in developing tissues that give rise to the vast majority of tumors that affect children and young adults, medulloblastoma and neuroblastomas, Wilms tumors and other kidney tumors, sarcomas, and a variety of gliomas. And so today I'd like to explore with you the idea of other developmental oncogenic mutators like RAG1/2 that are part of normal physiologic healthy development that become dysregulated to make mutations that ultimately cause to childhood cancer. Now, this idea is not so far-fetched because if we look at the genomes of cancer cells, and here is a karyotype of the earliest published childhood cancer genome, a neuroblastoma from a toddler, published by Spriggs and colleagues in the 60s. Here we recognize, of course, one of the hallmarks of high-risk neuroblastoma, which is that in addition to these normal chromosomes that are part of our normal healthy cells, these neuroblastoma cells also have numerous small so-called double minute chromosomes that in this particular case lead to the overexpression of the MYC-N oncogene and cause the hallmarks of high-risk neuroblastoma. The process by which these chromosomal abnormalities occur in healthy developing cells remains a mystery to this day. And I would posit to you that we need new ideas, such as developmental mutators, to explain their development. The same is true for many, many other tumors that affect young children. Here's, for example, a karyotype of rhabdoid tumors that I'll spend some time talking about. This is from Delacha Arias and colleagues. And here, in this tumor from an otherwise healthy infant, they recognize the presence of a chromosomal translocation from chromosome 1 to chromosome 22 that leads to the inactivation of the SMARCB1 tumor suppressor gene, indeed now recognized as the pathognomonic feature of the majority of rhabdoid tumors. How this chromosomal translocation occurs in an otherwise healthy infant, and these cells that have not had that many replicative cycles of division have not been exposed to mutagens in the environment, is a mystery that I'd like to explore today and suggest the existence of a developmental mutator that's responsible for making this mutation. Now, this work originally began some number of years ago with a simple idea that if the genomes of cancer cells are complex and are cryptic with respect to the mutational processes, then perhaps we could look at the proteomes, the output, the protein output of these tumor cells using new methods in mass spectrometry proteomics. And that's indeed what we did over many, many years. One aspect of this work, initially focused on childhood kidney tumors, was published by Michael Ortiz and colleagues recently from my lab, along with Anton Hensen from his own lab, that discovered a new mechanism of chemotherapy resistance in childhood solid tumors having to do with the overexpression of the molecule prohibinin that regulates the apoptotic mitochondrial signaling. Very interesting work that I would like to highlight here to draw your attention so you could explore later when you have time. But as part of this, coincidentally, in this work, we also observed in rhabdoid tumors, childhood rhabdoid tumors, a very highly expressed, at the time, orphan human gene that called PGBD5. It stands for piggyback transposable element derived gene number 5. Very peculiar name that caught my eye because at the time, the piggyback class of DNA transposases that I'll tell you in a bit was re-engineered and repurposed for gene transfer technology and transgenesis. And it seemed odd that a gene related to this otherwise very powerful enzyme that can mediate gene rearrangements would be expressed in these otherwise genetically quiet developmental tumors. And so before I go on, I want to spend a few minutes giving you some background on transposable elements and transposons. This is a fascinating class of genetic elements that's present in virtually every living thing, from prokaryotes to eukaryotes to animals. Originally discovered by Barbara McClintock in corn, and in fact, these are the 2 corn cobs that she studied, that Rob Bartnison gave me to hold for a few minutes. She was studying a very fundamental developmental process in plants, which has to do with color variegation and the production of differently pigmented corn kernels on this corn cob. This is a developmental phenomenon in plants that's important for development and stress response. And what Barbara McClintock found is that the production of this pigmentation gene and the pigment is controlled by DNA rearrangement, by a DNA transposase called activator and dissociator, that leads to change in the gene expression through mutations that are developmentally encoded. And since then, a variety of investigators and fields have recognized that these genetic elements, genes that can mediate somatic DNA rearrangements, are actually spread throughout all living things. In yeast, the CAT1 transposase controls the mating switch. In ciliates, the piggyback transposase controls development from the somatic to the germ cell nucleus. In plants, I already told you about activator from Barbara McClintock's work, but there are many, many other transposable elements important for development and evolution and stress response. In insects, you may have heard of the P element in Drosophila, which is a transposable element that by accident invaded laboratory strains and then was repurposed for forward genetic screens. The piggyback transposase was discovered by Malcolm Fraser and colleagues in the looper moth, as I'll explain in a second, that mediates DNA transposition also. And of course, over the years, the field has wondered whether there are, as of yet to be discovered, vertebrate transposons. And as I'll show you, the answer is there are many, and they're fascinating, and at least some of them, like PGBD5, contribute to childhood cancer and human disease. Now, why this focus on transposons? The answer, again, lies in very simple observations, which is that if you look at the human genome, and here are three famous human cancer genes, where these vertical marks represent their coding exons, and these intervening sequences represent the introns. If you look at inside the introns, there's a whole universe of sequences derived from transposable elements. Some of them are RNA mediated, some of them are DNA mediated, but suffice it to say that almost half of the human genome by weight is derived from transposed and transposable elements. Of course, most of them are evolutionarily extinct. They're vestigial structures that we carry by virtue of our evolutionary history, but some of them are actually quite important and functional and at times pathogenic. Now, PGBD5 caught our attention for the simple reason that it's not only expressed in the majority of rhabdoid tumors, it's expressed in the majority of childhood medulloblastomas, neuroblastomas, ependymomas, Ewing sarcomas, and other fusion-positive sarcomas like the chromosomally translocated rhabdomyosarcomas, but not many other tumors like the leukemias where REC1 is expressed. And PGBD5 is also compelling as a developmental mutator because it's deeply evolutionarily conserved among vertebrates. Here is its phylogeny, where it's highly, highly conserved among humans, rodents, fish, all the way to the ancestors of the bony fishes, divergent from the insect piggyback. The way these genes work is that the transposase itself encodes an enzyme, a protein enzyme, that in a sequence-specific manner recognizes specific sequence elements in the DNA and leads to their excision, followed by insertion in this cut and paste-like reaction, where the insertion target sites have characteristic TTAA target site duplications. And that's how, for example, the piggyback gene spreads itself through the insects. And that's how piggyback was repurposed for transgenesis, where the transposon cargo is not natural, but synthetic to insert genes into cells. It turns out that the human PGBD5 works in many ways just like the insect one, in that it can mediate the same cut and paste reaction in human cells. And Anton Hensen, when he was in my lab, led this work and mapped the integration sites of transposons mobilized by human PGBD5 in human cells, and found that they were integrated in all the chromosomes on the circus plot, from the chromosome 1 to all the small chromosomes, into genes and between genes, with characteristic target site duplications and a sequence-specific activity that's almost indistinguishable from the piggyback transposase. So PGBD5, human PGBD5, is truly an endogenous human DNA transposase. Now, this, of course, led to a very provocative hypothesis that I outlined here, that we term the developmental mutator hypothesis. It has, in spite of being very provocative, it has very, very testable predictions. The first of which is that the transient expression of PGBD5 should be sufficient to heritably transform human cells by virtue of causing mutations in the DNA that are, of course, heritable. That the enzymatic activity of PGBD5 should be required for cell transformation. And that PGBD5-induced transformation should induce specific genomic rearrangements with specific sequences that are dependent on specific targeting and DNA repair factors. And indeed, in work that Anton published a number of years ago, we tested each of these predictions, and I invite you to read this paper in detail, as it's quite comprehensive and describes in detail the fulfillment of each of these predictions by PGBD5, confirming that it's a developmental mutator. Here, I'll just show you some key examples of this work. In one set of experiments, we took primary human cells that are themselves genomically stable and do not transform spontaneously. And we expressed human PGBD5 under the control of a doxycycline-inducible promoter, and then transplanted these cells into immunodeficient mice, where, in the presence of doxycycline and induction of PGBD5, these cells become transformed and form tumors, as shown on these volumetric measurements. If you withdraw doxycycline from the diet of these mice, leading to the loss of all measurable PGBD5 protein expression, as shown in this Western blot, the tumors nonetheless continue to grow just like their counterparts. And if you transplant these tumors into totally naive mice that have never been exposed to doxycycline, they nonetheless continue to grow with a morphology that's indistinguishable from their counterparts, indicating that transient expression, just for a few weeks, of normal human cells with PGBD5 is fully sufficient to lead to fully penetrant, serially transplantable tumors. This process requires the enzymatic activity of PGBD5, where the enzyme uses a triad of 3 aspartic acids to catalyze magnesium-dependent hydrolysis of DNA and mediate DNA transposition, where mutants from aspartic acid to alanine that are now deficient in enzymatic activity as measured by this transposition assay using quantitative PCR, these mutants also become inactive for cell transformation, as shown in these survival plots of mice, immunodeficient mice, that have been engrafted with these tumor cells, where wild-type PGBD5 causes fully penetrant tumor formation in just 3 weeks, whereas these catalytically inactive mutants are unable to transform cells in spite of being expressed just as well, and in spite of localizing to chromatin almost just as well as wild-type parts. There's one exception, which is this mutant, which is worth discussing. So if you have questions, please let me know. We can discuss. Very interesting exception to this rule that doesn't disprove the hypothesis, just a caveat. Now, these transformed cells are transformed by virtue of somatic DNA rearrangements. And here is one example that we found, which is a duplication of a tumor suppressor gene called WWOX, here confirmed by cloning of the duplication breakpoint and Sanger sequencing. And remarkably, this tandem duplication leads to the evolution of an entirely new mutant gene, here shown on this Western blot, where normal WWX is 40-some kilodaltons, but this duplicated one is extended by virtue of this duplication and leads to the expression of a dominant negative form, mutant form, that's required for cell transformation. And this occurs not just in a singleton fashion, it occurs in multiple independently initiated tumors, consistent with the idea that PGBD5 targets specific sequences in the human genome in a programmed manner that recurrently leads to stereotypical DNA rearrangements, at least some of which are mutagenic, leading to cell transformation. And indeed, if you look genome-wide by whole genome sequencing at these tumors, as Anton did with Richard Koche and colleagues, here shown in the circus plot from the big chromosomes all the way to the small chromosomes, we find a variety of, again, somatic mutations. Most of them are deletions, but sometimes translocations, duplications, and inversions. And a large fraction of these mutations induced by PGBD5 at their breakpoints, as marked by these arrowheads and shown here in these sequence alignments, have a very characteristic sequence. If you read it from the right to the left, it's GTGAC, 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 and so on, which we termed PGBD5 specific signal sequence by analogy to the RAG1 signal sequence that are the substrates for the RAG1 recombinase, leading to the suggestion that these are some of the sequence elements in the human genome that are recognized by PGBD5 to mediate their somatic rearrangements. Again, in this case, mostly deletions and translocations. And you would then predict that if this is true, then if you look at human tumor cells, like, for example, human rhabdoid tumor cells that express endogenous PGBD5, where we, of course, hypothesize that it's responsible for the somatic DNA rearrangements that lead to mutations that lead to rhabdoid tumor development, that PGBD5 in these tumor cells would be physically associated with some of these genomic elements in the human genome. And indeed, using chromatin immunoprecipitation and sequencing, we mapped the genomic distribution of PGBD5 transposase on human chromatin. And we find that inside these regions bound by PGBD5, there's a very strong enrichment of sequences that look similar to the PGBD5 signal sequence, the GTGAC motif that I just showed you, but not their scrambled counterparts, nor the RAG1 signal sequences, which are, of course, irrelevant in this cell type. And you would also then predict that some of these human sequences would be direct substrates of PGBD5. So to test this, we used a reporter system initially developed by Malcolm Fraser and Nancy Craig and colleagues, where a reporter gene, in this case, a neomycin's resistance gene or a fluorescence gene, are flanked by these sequence elements. In one case, for example, these are, in this blue, are the insect transposons that the piggyback transposase works on, or these human elements, or of course its control counterpart that just has the reporter gene but no transposons. And if we express these or introduce these synthetic substrates into human cells expressing either PGBD5 or controls, we could then estimate the transposition activity by counting the number of cells that emerge to be neomycin resistant after the transposition reaction. And so, of course, if you take normal human cells, they are not resistant to neomycin. They do not produce any colonies, regardless of whether they contain insect transposons or human transposons. If these cells express the insect transposase, it efficiently transposes and integrates the neomycin resistance gene into the human genome, leading to the production of resistant colonies. And remarkably, PGBD5, human PGBD5, does the same thing. It mobilizes both the insect transposons in these new human sequences, consistent with this hypothesis that it is a developmentally encoded somatic mutator that can make DNA rearrangements in human cells. And remarkably, if you look at these cells and again map the distribution of PGBD5 protein on the human genome, here, for example, is a representative locus on chromosome 1, where we find the concentration of PGBD5 in association with this PSS motif that I just mentioned, the GTGAC. We also find at exactly the same position integration of these synthetic transposons, suggesting that PGBD5 exists in human tumor cells in a poised state that's competent for DNA transposition and DNA rearrangements, consistent with this mutagenic activity that I described to you. Does this happen in human tumor cells? We began this work in rhabdoid tumors, and so together with the Target Consortium and Elizabeth Perlman and many colleagues, we examined human rhabdoid tumors isolated from infants with tumors and sequenced by whole genome sequencing as compared to their matched normal tissues. And here is an analysis of this data that again was published by Anton and Richard a number of years ago, showing you all the human chromosomes from the big to the small on this circus plot, where we have overlaid all 30-some tumor genomes on one circus plot to show you the patterns of somatic DNA rearrangements that occur in these rhabdoid tumors that are recurrent in multiple individual patients. And so here, for example, you will see that on chromosome 22, virtually all patients, all tumors, have recurrent mutations of the SMARCB1 locus that inactivates the SMARCB1 tumor suppressor, leading to rhabdoid tumorigenesis. Most of these events are deletions, but some of them, if you will notice, are also translocations that are coupled to the deletion event, meaning the mutation itself is a complex DNA rearrangement. And you will see that across the genome, there is a variety of these coupled rearrangements, deletions, translocations, inversions, and that a large fraction of them at their breakpoints, as indicated by these arrowheads, contain the same characteristic signal sequence, GTCAC. The PGBD5 signal sequence that I just showed you is induced by PGBD5 in normal human cells. Here's a slightly more detailed view of this, where I'm showing you the various genes that are recurrently mutated in individual tumors from individual patients. Many of them, of course, are SMARCB1, but there are many others like this contactin. And again, at or near the mutation breakpoint is the same sequence, GTGAC, 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 again, the PSS sequence. The sequence motif is imprecisely positioned relative to the breakpoint, consistent with a slightly promiscuous activity of PGBD5. In other words, an aberrant activity, not a perfect cut and paste reaction, but maybe imperfect excision event, or perhaps just a double strand break that then gets repaired inappropriately to produce these deletions. Now, as we finished this work, it was very gratifying to see that several other consortia, Stefan Pfister and Marcel Kuhl, Annie Huang, and Tamar Jabato completed their own genome sequencing analysis of rhabdoid tumors, both kidney tumors and rhabdoid brain tumors, the ATRT tumors. And it turns out from this body of work, we now recognize that rhabdoid tumors actually exist in at least 2 or 3 distinct molecular subtypes here in the Pfister and Kuhl classification, the tyrosinase, hedgehog, and MYC subtypes, and that at least in part, the differences in the molecular properties of these different rhabdoid tumors are associated with differences in the types of mutations that affect the SMARCB1 locus and other linked events, consistent with the idea that rhabdoid tumors are produced by a somatic mutational process that leads to their diversification, and in this particular case, differences in patient outcomes. Now, this was, of course, very exciting to us, but looking back, it turns out that actually there's been already a lot of ample evidence to indicate this. Here's, for example, I'm showing you deletions of the contactin gene, which is recurrently mutated in rhabdoid tumors that with the breakpoints that we cloned in Sanger sequence that I'm showing you here. But it turns out that recurrent deletions of the contactin gene on chromosome 7 were already reported here, for example, by Ogawa and colleagues using CGH arrays a number of years back. So in other words, SMARCB1 is indeed an important part of rhabdoid tumorigenesis, but it's not the only one. And based on our analysis, on average, each rhabdoid tumor has approximately 3 to 4 additional mutations besides SMARCB1. Often this contactin gene is deleted. Again, consistent with the idea that childhood tumors, even if they're quote unquote as relatively simple genomically as rhabdoid tumors, are nonetheless products of somatic mutagenesis and tumor evolution. And so this comes back to this idea that I raised to you a few minutes ago, which is PGBD5 a developmental mutator that's dysregulated in developing tissues in healthy children that then leads to the initiation of the various childhood solid tumors where PGBD5 is expressed in the majority of rhabdoid tumors, medulloblastomas, fusion sarcomas, and neuroblastomas. Now, this question, of course, is difficult to address decisively from human studies. But luckily, over the years, the field has developed a variety of genetically engineered mouse models that are quite accurate developmentally. And so we turn to these mouse models to ask whether PGBD5 is required for the initiation of tumor development in mice, insofar as these mouse models have produced the human condition. So we've been working together with several colleagues to address these questions across these many tumor types in mice and children. And the first one that I want to present to you, and this is unpublished work, is in medulloblastoma, where initially we collaborated with Michael Taylor's group and Hiro Suzuki and his group to examine human medulloblastoma genomes and to specifically ask whether in the somatic mutations that characterize human medulloblastomas, whether the breakpoints of these mutations have specific sequences consistent with the existence of a specific mutational process, and whether these sequences are in fact PGBD5 signal sequences that we just found in rhabdoid tumors. So in this work, we, together with Michael, we examined several hundred cases, initially of hedgehog subtype medulloblastoma, but now all the 4 major subgroups. These Illumina genomes were analyzed by Deli and Geneman, and then importantly, the reads that are rearranged were locally assembled using Velvet, giving us the contigs that we could then examine for sequences. And so here's a summary of this work that Ross Keller and my lab compiled together with Hiro and Michael's group, where we're showing you the various genes that are recurrently mutated in multiple individual medulloblastoma genomes. And if you look at these genes, and of course you will recognize if you've been studying medulloblastoma that many of these are well recognized tumor suppressor genes or oncogenes in medulloblastoma, genes like GLI, SUFO, and PACHT1 that encode factors in hedgehog signaling, genes like MIC-N and MIC, of course, that are important pathogens in a variety of tumors, genes like JFI1, 1B, PPM1D, and BRDM6. And importantly, in nearly a third to a half of these somatic mutations, deletions, inversions, translocations, we found that their breakpoints have specific sequences at their breakpoints, as opposed to just random sequences that are characteristic of the human genome at large. And here's a breakdown of this analysis where I'm showing you here the various subgroups, the hedgehog subgroup, group 3, or MIC activator subgroup, group 4, WNT, and in comparison, genomes from breast carcinomas, from patients with tumors that are deficient in homologous recombination that produces a whole universe of somatic mutations that are highly complex as a kind of comparator to give us a sense whether the signals we're seeing at the breakpoints in medulloblastoma mutations are true or just byproducts of somatic mutagenesis that may happen during tumor development. And importantly, we found that in, you could see 10 to 20% of the tumors, we find that their breakpoints look very much like the GTGAC motif that I just showed you that characterizes mutations in rhabdoid tumors and PGBD5-induced RPE tumors, but not in these complex breast carcinomas. And these medulloblastomas, in addition to the PGBD5 signal sequences we already defined as substrates PGBD5, also have several other new sequence motifs that, again, as I mentioned, in aggregate account for a third to a half of somatic mutations in human medulloblastomas, consistent with the existence of a specific mutational process that leads to, again, mutations of tumor suppressor genes and activation of oncogenes that are responsible for human medulloblastoma. Is PGBD5 responsible? Again, as I mentioned, in human studies, it's very difficult to answer this question decisively, but in mouse studies, it's very tractable. So we collaborated with Praveen Raju and more recently with Alexander Joyner, who have developed a variety of developmentally accurate models of hedgehog signaling medulloblastoma. Here, for example, is a model that uses somatic inactivation of the patched1 gene, which is a negative regulator of hedgehog signaling, with deletion of patched1, leading to constitutive activation of hedgehog signaling in developing cerebellar progenitor neurons in developing mice, on average several hundred such initiating events per mouse cerebellum, leading to, over several weeks to months, development of fully penetrant medulloblastomas that ultimately kill all mice that are engineered this way, consistent with, again, what happens in human medulloblastomas with activation of hedgehog signaling. And remarkably, whereas mice that are wild type for PGBD5 develop these tumors rapidly, mice that are littermates that are deficient for PGBD5 are in large part protected for a long, long time. As you can see, the majority of these mice do not develop tumors for almost a year, indicating that mouse PGBD5 in developing cerebellar progenitor cells is required for the initiation of otherwise fully penetrant and lethal human medulloblastoma, consistent with the function of PGBD5 as a developmental mutator that I just showed you. Now, this, of course, raises a question that I'm sure some of you have been thinking about, that I have been thinking about for many years, that people always ask, which is, if PGBD5 is doing this abnormally in developing tumors, what is its normal function? And I can't spend much more than a few minutes on this question here because it's far too expensive for this talk, but suffice it to say that DNA rearrangements in neuronal tissues, developing neurons, have been hypothesized for a long time, over 50 years, for a variety of reasons based on a variety of observations. And of course, we can't help but ask whether PGBD5 is the elusive neuronal DNA recombinase because of the existence of these mouse models that we have developed. And so Luce Jubier and colleagues in my lab asked this, looked at the developing mouse neurons. Here's a section through the cortex of a developing mouse embryo. This is the ventricle, and this is the outside of the brain. And it's been recognized for a long time that these developing mouse neurons normally undergo a variety of double-strand breaks measured using NMIC labeling with tunnel, and also captured by the activation of DNA damage repair by phosphorylation of histone 2AX here in red. You could see that a variety of double-strand breaks and neurons with activated DNA damage. And remarkably, mice that are deficient in PGBD5 have a very significant and reproducible reduction in the amount of double-strand breaks in developing neurons, consistent with the activity of PGBD5 during normal development that somehow is dysregulated during tumor development. To explore this further, we carried out a relatively large study of the genome sequencing of normal developing mouse brains, both from the adults, from different regions in adults, and the embryos. Importantly, together with a variety of colleagues, Raoul Rabadan, David Torrents, and many others, tried to apply new methods for the analysis of short-read DNA sequencing that are able to capture what would otherwise be polyclonal somatic DNA rearrangements. And indeed, here's an example of the success of these new methods where we, in normal blood of mice, can now detect deletions in the immunoglobulin kappa locus, which encodes one of the immunoglobulin genes that are normally rearranged by the RAG1 recombinase as part of normal B cell and T cell development that I just told you, that again produces these quote unquote mutant but normal healthy B cells and T cells in the circulation. And in the brains, we see also a large spectrum of somatic DNA rearrangements that occur in wild type brains, but not PGBD5. Knockout brains, these are concentrated in distinct regions of the mouse genome, consistent with a specific activity. And here is just on this heat map, I'm showing you some of the loci that are, again, somatically rearranged in wild type mice, but not in mice that are deficient for PGBD5. And we're currently actively trying to confirm and establish this phenomenon rigorously. We hypothesize that if PGBD5 is physiologically important, as its evolutionary history suggests, as its mouse knockout phenotype indicates, that PGBD5 would be working either like RAG1, in which case it would mediate somatic DNA rearrangements of specific genes, leading to their somatic diversification, or alternatively, more like Barbara McClintock's activator, where it would make DNA rearrangements to control gene expression. And so I hope in the next year or two, we'll be able to definitively answer the question of what is the function of PGBD5 in normal physiologic development, and of course, by extension, how it may be dysregulated in developing tumors to produce mutations that are characteristic of childhood solid tumors that I just mentioned to you about. So in summary, the model that we've currently produced and are trying to refine is that PGBD5 is associated physically with the human genome with specific sequences, at least some of which look like the PGBD5 signal sequences that I just showed you, together with cofactors, at least some of which is PAX, which I didn't have time to tell you about, but is published, mediate somatic DNA rearrangements that lead to mutations of oncogene and tumor suppressor genes that are, at least some of which are then pathogenic and are required and sufficient to transform cells and mutate them. Now, this was again a lot of ideas. And as you would imagine, this biology must be complex because such a process must be tightly regulated in our cells. And so we have a lot of work ahead of us to enumerate the factors that are working with PGBD5 and to define their mechanisms. But nonetheless, we, of course, as pediatric oncologists are very eager to try to apply knowledge of this to new therapies for patients. And so one idea that we explored early on is that if PGBD5 is active in tumor cells and makes double-strand breaks and other forms of DNA damage, then perhaps these tumor cells would require ongoing DNA damage repair and signaling to repair many of these events that PGBD5 induces, as I showed you. And so this work, again, was led by Alex Kentsis when he was in my lab, where we explored the dependency of PGBD5 on non-homologous and joining DNA repair, because this form of DNA repair is classically engaged by DNA transposases like PGBD5. This involves the structural cofactors like the Ku factors that load onto broken double-strand ends and recruit a large apparatus, including the ATM and ATR kinases that signal to remodel chromatin, recruit DNA ligases, and ultimately repair the break. And what we found, again, in work that was mostly published, is that PGBD5 expressing cells require non-homologous end joining. They require both Ku and ATM and ATR as measured by NIC labeling and phosphorylation of H2AX. And if we survey at the time existing selective inhibitors of DNA damage repair, like this ATR inhibitor, ATM inhibitor, and the like, and compare their activity against cells that express wild-type PGBD5 or the catalytically inactive and various other mutants, we could identify inhibitors like this ATP-competitive ATR selective inhibitor AZD6738 that are more than a log selective, again, against PGBD5 expressing cells, but not normal cells. Importantly, this inhibitor exhibits a very high potency in vitro against the majority of rhabdoid tumor cell lines, medulloblastoma cell lines, neuroblastoma cell lines, Ewing sarcoma cell lines, all of which express PGBD5, but much reduced activity against normal human and mouse cells. And indeed, to a first approximation in vitro, the susceptibility of human PGBD5 expressing childhood tumor cells to AZD6738 correlates very well to the relative expression of PGBD5 protein. Not perfectly. In other words, there are other determinants of this, but as you can see, at least half of the variation is explained by PGBD5 expression, consistent with the idea that PGBD5 is both necessary and sufficient to confer this dependency, which we confirm genetically, where we take rhabdoid tumor cells, deplete endogenous PGBD5 using multiple independent RNA interference constructs, and then compare these PGBD5 proficient tumor cells as compared to their PGBD5 deficient counterparts, and find that depletion of PGBD5 confers relative resistance to this ATR inhibitor, consistent with the idea that it's required for this particular susceptibility and a kind of a synthetic lethality, if you will, in pharmacologic terms. And gratifyingly, some, but not all, human tumor cells that express PGBD5 could be significantly suppressed in their growth and sometimes regressed by single agent oral ATR inhibitor treatment, as shown here, for example, for these two neuroblastoma and medulloblastoma xenografts in immunodeficient mice. And this leads to, again, this idea that PGBD5 in tumor cells remains active, continues to make mutations and damage DNA, and this requires ongoing DNA damage repair signaling, repair and signaling, where inhibition of the specific pathways that are engaged by PGBD5 could impair this DNA repair, leading to reduced cell survival and cell death. And so we are working hard to understand the molecular mechanisms of this oncogenic form of DNA damage signaling. Of course, we're working hard, as funded by the Alex Lemonade, to develop rational strategies for targeted interference with this, and actually immune therapies that I didn't talk to you about today that are derived from PGBD5 activity. And satisfyingly, Michael Ortiz, based on this work, is planning to open, together with the COG and Bayer, a new clinical trial this fall using the new ATR selective inhibitor to, again, test this hypothesis in children with recurrent or refractory solid tumors, most of which express and activate PGBD5. And so in the last few minutes, I just want to conclude and ask a more broader forward-looking question, which again concerns this idea that our tissues and our genomes are not static, that they are a product of development, and at least some of this development involves somatic DNA rearrangements, some of which are physiologic and developmentally regulated, which would explain the incidence of mutations in otherwise healthy developing tissues. And so this question of could there be somatic genetic diversification by domestic DNA transposases is actually, though at the moment seems very provocative, but it's actually not very new, because even when the human genome was completed in the late 90s, Arian Smith and colleagues noted that the human genome contains a variety of genes that are derived from transposons of various types. Here is the reproduction of their table from the late 90s. You will see many of these genes here have unusual names because they actually remain orphan in that they have not been studied. PGBD5 is not on this list. And so suffice it to say, there are probably several dozen genes that are in human genomes, some of which are evolutionary conserved, some of which retain apparent catalytic residues, and are expressed in tissues. And so it raises the possibility that perhaps our tissues, like the tissues of many other organisms, are somatically diversified. We talked about REG1 and 2. That's, of course, essential for adaptive immunity. And as I mentioned to you, is the principal driver of lymphoid malignancies, leukemias and lymphomas in both children and adults. We talked about PGBD5 that I hope I've convinced you needs to be explored further as a developmental mutator responsible for the initiation and diversification of the majority of childhood solid tumors and probably in normal neuronal development. There are many others like FAP9, which is a P-element transposase discovered by Don Rio that's active in human cells, SETMAR, HARB1. Recently, topoisomerase has been implicated in nuclease-mediated rearrangement. So there's a lot of biology that we need to work on. And finally, of course, I want to leave you with the idea that this work and work of many others suggests that childhood cancer needs to be thought of as a disorder of developmental somatic DNA rearrangements, where normal and healthy progenitor and stem cells undergo somatic DNA rearrangements, at least some of which are mediated by PGBD5 and RAG1, and this process leads to the inactivation of tumor suppressor genes and activation of oncogenes that characterize all the features of childhood and young adulthood somatic tumor development. This is the extension of the developmental mutator hypothesis that Steve Frank and I formalized earlier this year in a paper cited here. And of course, it also raises questions and provides a very satisfying framework of how to think of why do young people get cancer in the first place. And in this work that I cite here, I'd like to present you with this simple framework, which is that the reason why young people get cancer has to do with the interaction of constitutional cancer predisposition that we now know affects at least 10% of children and adults and maybe more. It also involves exposures and oncogenic pathogens that could facilitate the acquisition of mutations in response to these predisposing genetic mutations. And of course, it involves developmental mutators, or in other words, processes that are inherent to our normal cells that are important for normal physiology and normal development and normal homeostasis that can mediate somatic DNA mutations, either from nucleases like PGBD5 and RAG1, or perhaps mitotic processes having to do with cell division. And together, these three factors and the interaction among these three factors is responsible for the onset of cancer in young children and adults. And so with that, I think I am on time, and I want to finish as I have tried to acknowledge along the way. This work is highly collaborative and is a product of extensive work by many, 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 many people. I've tried to cite some of the key contributors, but the rest of them are listed here. There are many, I'm sure, who are listening to me now, and I thank all of them. And with that, I thank you. I thank Jay and Alex's Lemonade, and I will take your questions.
Thank you so much, Alex. That was very informative. Certainly looked at cancer from a different perspective than the lectures we've heard up until this point. And it's not something that I've heard before. So I really appreciate that. I'd like to give people a chance to type in questions into the chat box and we can have them answered. So we'll just give them a minute. Type some in. Hopefully we get a few. Okay, first one coming in. Alex, great talk. You may have answered this question. Fortunately, rhabdoid and other pediatric brain tumors in children in the brain is relatively rare. If PGBD5 is expressed in all neuronal cells, can you comment on how PGBD5 function is regulated, dysregulated, and possibly suppressed to prevent somatic mutagenesis and oncogenesis?
Yes, that's right. I think that this is the key point, is that fortunately, tumors that affect children are relatively rare. This includes rhabdoid tumors and medulloblastomas and many others. And so insofar as PGBD5 is expressed in all of these cells, in all of us, it must be tightly regulated. And we, of course, hypothesize both negative and positive regulators that control its activity during specific developmental periods and prevent the unrestricted tumor mutagenesis that can accompany tumor formation. To be honest, we still don't know what they are, but I would posit that at least some of them are factors that regulate DNA damage repair itself. And of course, it's very tempting to postulate that the reason why in some children PGBD5 causes mutations that leads to tumors like medulloblastoma and rhabdotumors and the like, is that these individuals have an underlying slight defect in DNA damage repair. And so, for example, an allele, a hypomorphic allele in ATM or ATR or the like, that normally is fully competent and fully functional and allows for efficient DNA repair, but in cells where PGBD5 is activated is inadequate. And so we're actually currently looking in two large whole genome studies looking at the germline genomes of children and their parents to try to identify these predisposing alleles. So I hope that answers your question.
Thank you. That came in from Martine Rousseau.
Oh, well, thank you, Martine.
The next question is from Shai Israeli.
Jay,
Okay.
I lost you there for a second. Can you repeat?
Great talk, Alex. A truly inspiring vision. The question is the expression of this transpose, most of adult sarcomas have also have specific.
Yes, Jay, if I
Sure.
heard you correctly, the question was whether PGBD5 is also expressed. Yes, is expressed in adult sarcomas. Yes, go ahead. No, but not anymore.
What about sarcomas? Because they have specific fusion genes.
Yes, that's exactly right. We are actively looking
Maybe Jen
at...
could read it.
Go ahead, Jennifer.
Okay, I think you got that question. If you want to answer that one, and then I'll read the remaining ones.
Okay, good. So if I got the question, is whether PGBD5 is also active in adult sarcomas. And the answer is we hypothesize yes. For example, in the fusion positive sarcomas, like fusion positive rhabdomyosarcomas, for example, we also hypothesize the same. And we have some unpublished data that is consistent with that, where we've looked at the composition of the sequence breakpoints in these sarcomas that look like PGBD5 signal sequences. And of course, we're actively collaborating to test this in mouse models, for example, using the very nice mouse models for rhabdomyosarcoma that many have developed, including at St. Jude. And the same is true actually for carcinomas, because PGBD5 is also expressed in the majority of small cell carcinomas in young adults and middle-aged adults, small cell lung cancer, small cell colon cancer, small cell breast cancer. And so we're exploring the same in that. And of course, that's very interesting because the genomes of these small cell carcinomas are highly complex, highly mutated. And so we're looking for that.
All right, so Alex, I'm going to try and do this without my video on. Maybe my slow internet connection today will work, okay? The last slide, this question is reference to the last slide. Predispositions were mentioned as 10% of causes of pediatric cancer. Do we know the percentages for the other two groups?
For the other two groups, meaning the pathogens and mutators, or which other two groups?
That was the entirety of that question.
Oh, well, so, I presume it relates, so the 10% estimate comes from several studies of both children and adults. It's an approximate estimate. The numbers vary, of course, by...
It must be the oncogenic pathogens and developmental mutations.
Yes, right. So that estimate varies, and in some groups and tumors, it's higher, in some it's lower. Now, as far as the oncogenic pathogens or exposures, there are, of course, clear cases where that's the case. For example, human papillomavirus and the causal relationship between human papillomavirus and probably half of all oropharyngeal cancers in young adults. But in many other tumors, we don't know. They've been hypothesized based on epidemiology and incidence type of analyses, but no one has yet found oncogenic pathogen or an exposure in these other cases. But of course, people are looking, and for example, recently work from Matthew Meyerson has nominated a specific type of fusobacteria in the colon as a contributor to the pathogenesis of a distinct subset of colon carcinomas. And so this is a very, very active field, and I fully anticipate that over the years to come, we will find many other external causes of human cancer, be they pathogen, viruses, or exposures. And so the challenge will be actually, even though this diagram that I show here is very simple, the challenge actually is very, very daunting, how to understand the relationship among these three things, because epidemiology becomes very, very complicated when you need large cohorts, and model studies in animals need to be very, very refined and very, very sophisticated to incorporate accurately all of these factors. So this will be a fueling, I think, a lot of effort over the many years.
Great, thank you. This question comes from Michael Wiggins. Very basic question. As a pediatric cancer parent, I'm often asked whether cancer in children is genetic or hereditary. Is there a difference? And if so, can you explain?
It's a great question. Of course, and there is a difference. Of course, we know that for some children, they develop cancer in a hereditary fashion where they inherit altered form of a gene from either the mother or the father, and that this altered form of a gene runs through the family in a hereditary fashion. And so these are some of the well-recognized cancer predisposition syndromes. But we also know of cases where we do not see such an altered, but instead see it originating in the child, him or herself, during early development. Some of these altered forms are constitutional, where the entire body has this form. Some are mosaic, where certain tissues have this form. And so again, these are relatively rare events. The numbers are relatively rare, 10% or so. But the idea that I would like to explore here today is that perhaps there is another process that's not hereditary, because the altered forms of genes don't run in families, but instead it occurs in the body, in specific tissues in the body that then lead to tumor development. So in that sense, it is a genetic cause, but it is not a hereditary cause. And so this idea that now is increasingly gaining traction in the field, because as we analyze the genomes of normal tissues in children and adults, not from children with cancer, but just otherwise healthy children, we find that their tissues actually have lots and lots of altered genes in them. Most of them are probably not pathogenic and cause no harm, but some of them probably do. And that's the challenge for us, is to figure out what and how to leverage this knowledge for better therapies for all children.
Okay, thank you. We have another question from Logan Spector. If you have a couple more minutes, so just a couple more questions.
Of course, of course.
Okay, incidence of many childhood tumors vary by ancestry, most notoriously Ewing sarcoma. How does that fit with your hypothesis?
It's again a great question, and it comes back to the question that I think Martine asked earlier, which is, if PGBD5 is in all of us and is active in all of us, what dysregulates it in the first place? And of course, again, one attractive possibility is that perhaps there is an inherited gene that may be ancestry related as part of our ethnicity, historical ethnicity, or maybe just related to our family history, that is otherwise more or less normal, but in the setting of PGBD5 or another active developmental mutator confers an increased risk to having more mutations and special mutations that can cause cancer development in susceptible tissues. And so that's the question that we'd like to explore. Of course, this requires careful human genetic studies. And so that's the collaborations we're exploring now to try to understand exactly this relationship that is shown on this triangular diagram. Could there be an increased susceptibility to childhood cancer by virtue of our family history? That's not cancer predisposition syndrome, but just predilection, if you will.
Great. Okay, so it looks like this is probably our last question from Shai again. Is there any alteration of this transposon activity in children with ATM?
Yes, so great question. So Shai is referring to the ataxial telangiectasia syndrome, which manifests in children and young adults with blood cancers like lymphomas, and also neurodevelopmental defects, which is responsible for the ataxia and the problems with motor function that these individuals have. And we would hypothesize yes, and that this has to do with PGBD5 because, and this has been done extensively by many investigators. Martine Rousseau has done important work in this field. Fred Auld, McKinnon, many others. When they examined mice that are deficient for ATM, that mimic the ataxial telangiectasia of humans, if you look inside the developing brains of these ATM deficient mice, there is an excess of double-strand breaks and an excess in DNA damage repair activation and excess of neuronal cell death that's responsible for the abnormal development of these brains. And so that suggests the presence of a nuclease that's active in this period, which requires ongoing DNA damage repair and signaling, at least in part, dependent on ATM, the ATM kinase. And so that would be the hypothesis that we're exploring. And then again, that's very, very testable because of this preceding work that I just cited, and because the mouse models are currently very, very tractable. And so that I hope over the next year or two, we'll be able to answer this question. But my kind of intuitive answer today is that the answer will be yes.
Great. Well, thank you so much, Alex. Those are all the questions that came in. I know on behalf of Jay, he would also like to thank you for doing this. It looks like his internet booted him off early, unfortunately. But thank you all for attending. Thank you, Alex, for taking the time to present on your incredible work.
Thank you again. Thank you again. And I look forward to listening to more lectures in the coming months.
Thank you. Have a great day, everybody.